The Secret History of Fruits, Advanced Historical and Scientific Version. Long before the development of agriculture, early humans were not settled farmers. They were highly adaptive hunter-gatherers who relied entirely on environmental resources for survival. Modern archaeological and anthropological evidence suggests that wild fruits played a critical role in early human diets. Sites from the Paleolithic era show plant microfossils and seed residues, confirming that fruits were a consistent part of nutritional intake. Early humans did not understand microbiology or nutrition science. However, they practiced what scientists now call empirical food selection, a trial-and-error survival system. Fruits that provided energy without causing illness were remembered and repeatedly collected, while toxic or harmful fruits were avoided. Over thousands of years, this process created one of the earliest forms of natural biological selection influenced by human behavior. From a biological perspective, fruits were essential for early human survival because they provided three critical advantages. Rapid glucose-based energy for brain metabolism. Hydration through high water content. Micronutrients such as potassium, vitamin precursors, and antioxidants. These nutrients supported both cognitive evolution and physical endurance, especially in environments where food availability was unpredictable. The apple, a case study in ancient domestication. Genetic and archaeobotanical research confirms that the domestic apple, Malus domestica, originates from wild populations of Malus sieversii, still found in the Tian Shan mountain region of Central Asia. Fossil and genetic data suggest that apple domestication began more than 4,000 to 5,000 years ago along early trade networks such as the Silk Road. Early humans and traders unintentionally acted as selective agents by transporting seeds of sweeter and larger fruits across regions. A key biological property of apples contributed to their historical importance: post-harvest longevity. Compared to many soft fruits, apples have a relatively low respiration rate, allowing them to remain edible for extended periods under cool storage conditions. This made them especially valuable in pre-refrigeration societies for winter survival. However, apples also played a significant role in early food fermentation systems. Under natural conditions, sugars within apples can be converted by wild yeasts into ethanol, producing early forms of cider. Historical evidence from medieval Europe shows that fermented apple beverages were widely consumed due to safer hydration compared to untreated water sources. Conversely, improper storage conditions often led to fungal contamination, particularly by Penicillium species and other molds capable of producing mycotoxins. Without microbiological knowledge, ancient societies misattributed such illnesses to environmental or supernatural causes. Thus, apples function simultaneously as a survival food, a storage-dependent resource, a natural fermentation substrate, a hidden source of foodborne risk. The banana, a product of human-driven genetic engineering. Unlike apples, bananas represent one of the clearest examples of intentional human-mediated genetic selection. Wild ancestors of bananas contained large, hard seeds and minimal edible pulp. Archaeobotanical studies suggest that domestication began in Southeast Asia and Papua New Guinea over 7,000 years ago. Modern edible bananas, Musa acuminata and Musa balbigiana hybrids, are largely triploid sterile cultivars, meaning they cannot reproduce sexually. Instead, they are propagated vegetatively through cloned shoots. This makes bananas entirely dependent on human cultivation for survival. From a biological standpoint, this creates a major vulnerability: genetic uniformity. Because most commercial bananas are genetically identical, they are highly susceptible to disease outbreaks, as seen historically with Panama disease affecting large-scale plantations. Additionally, bananas exhibit a high ethylene sensitivity, causing rapid ripening. In tropical climates without controlled storage, enzymatic breakdown of starch into sugars leads to accelerated fermentation and spoilage. This made historical long-distance transport extremely difficult prior to modern refrigeration and controlled atmosphere shipping. The orange, ancient hybridization and nutritional revolution. Genomic studies show that modern sweet oranges, Citrus times sinensis, are not naturally occurring wild species but are the result of ancient hybridization, primarily between pomelo, Citrus maxima, and mandarin, Citrus reticulata. Selective cultivation in East and Southeast Asia over thousands of years gradually increased sugar concentration and reduced bitterness through human preference-driven selection. The historical significance of oranges became especially evident during the Age of Exploration, 15th to 18th centuries. Long naval voyages led to widespread outbreaks of scurvy, a disease caused by vitamin C deficiency resulting in impaired collagen synthesis. Although sailors did not understand vitamins, empirical observation showed that citrus consumption dramatically reduced mortality rates. Later scientific research identified ascorbic acid, vitamin C, as the key nutrient responsible for preventing scurvy. However, citrus fruits were highly perishable and sensitive to environmental conditions such as temperature and humidity, making preservation and transport a significant logistical challenge in maritime trade. The strawberry, a modern hybrid of fragile biology. The cultivated strawberry, Fragaria times ananassa, is one of the newest fruit species in agricultural history, originating in 18th century Europe through hybridization between North American and South American wild strawberry species. Unlike many fruits, strawberries are non-climacteric, meaning they do not continue ripening significantly after harvest. Their high water content, delicate epidermal structure, and enzymatic activity make them extremely susceptible to microbial spoilage. From a post-harvest biology perspective, strawberries have one of the shortest shelf lives among commercially cultivated fruits, which historically limited them to local consumption before the development of refrigeration and cold chain logistics. As a result, strawberries were historically considered luxury fruits, accessible only near cultivation regions or elite markets. Conclusion: fruits as a record of human evolution. When examined through archaeological, genetic, and biochemical lenses, fruits are not merely dietary items. They represent a continuous timeline of human interaction with biological systems. Early humans practiced empirical selection through survival-based observation. Agriculture introduced systematic domestication and selective breeding. Trade networks enabled global distribution and genetic mixing. Modern science decoded the molecular and evolutionary mechanisms behind these transformations. Fruits therefore represent a living archive of human evolution, connecting biology, history, and civilization. From wild forests to global supply chains, every fruit carries a hidden record of adaptation, selection, and survival. And even today, each bite reflects thousands of years of evolutionary biology and human innovation working together.